Okay. So the diagram shows a part of a cell at the start of metaphase 1 of meiosis. Okay. Which is organelle T. So what do you think is T?
Centriole.
Yeah, well done. It's correct. It's centriole. So what is the function of lysosome? So there are 4 answers, right? Now, A is centriole, B is lysosome, C is nucleolus, D is ribosome. Okay. What is the function of ribosome, Mitara? So, okay, let's discuss. Ribosome is the, so you know cell organelles, right? So when prokaryotic cells and eukaryotic cells, right? Prokaryotic cells are the cells that are, the bacteria is having prokaryotic cells. So all of us, the humans, plants, and all us, the genetically evolved organisms are having eukaryotic cells. So that means U means true. Eukaryotes means U means true. Okay. So we are having true cells. Okay. True cells contain organelles. Okay. True cells contain organelles. So these are few organelles in a cell. So let's find out the function of these organelles. Organelles means the parts of the cell. There are different parts of the cell which are specified for different actions. Okay. So likewise, these are 4 organelles. Lysosome. So then the name itself says lyse means destroy. So if we put an enzyme to a protein, what happens? The protein get lysed. Lysed means it is dissolving. That means it is getting destroyed. So lysosomes carries enzymes. It is a round shaped organelle, very small organelle in a cell. It contains different kinds of enzymes. To lyse them. So for an example, the protozoans, there are, amoeba, right? So the amoeba engulfs their food. Imagine this is the food and this is amoeba. And there are lysosomes here. Okay, there is a very small organelle called lysosome. It contains enzymes. Okay, so this food is engulfed into the cell, and there is this food. Okay, this is the food. Okay, hope you can see the screen very clearly. This is the food. So what lysosome does is it secretes lytic enzymes. Lytic means destroying enzymes, dissolving enzymes. So it can dissolve this food. So this food is broken down into small pieces and small elements by the enzymes of this lysosome. Likewise, lysosome in our body, do we have lysosomes? Yes. Lysosomes are used for cell apoptosis, cell death. So cell apoptosis means cell apoptosis. So when there is an unnecessary cell in our body, that cell is not unnecessary. Why that cell is unnecessary? So when cell division happens, Tara, This is the basement membrane. Imagine this is the basement membrane of our skin. Okay, so the cell division happens. These are different kinds of cells. These are the nuclei of these cells. Okay, so different kinds of cells are, the cell is dividing. So one cell is giving rise to several cells. Okay, so when cells are dividing, there can be genetic mutations, as you know. Okay, so there can be genetic mutations. So there can be either favorable genetic mutations or unfavorable genetic mutations. Okay, so imagine there is a very harmful or very absurd cell. The structure is very disoriented cell. In this cell making, in this cell division, there is a very unfavorable cell. So there can be favorable genetic mutations also. So what our body does is they apoptose these cells. We call that apoptose. Apoptose means our cell, our body itself destroy this cell. Okay, because this cell is very unfavorable, unhealthy, not doing its functions very well. So we have to destroy that. Okay, this cell apoptosis happens in a phase of cell cycle. There's a gene called P. You don't have to know all these things. Okay, there's a gene called P53. Okay, this gene acts as, you know what is a gene, right? Gene is when we take a chromosome like this is a chromatid. Okay, this chromatid contains a specific part which is responsible for a specific outcome. Okay, so imagine different people have different colored iris. The eyeball is different colors in different people. So one gene, or maybe there are several genes. For the gene, there is a gene which is responsible for the color of hair, the color of eyeball, the color of skin. Okay, that likewise, that gene is controlling that part. Likewise, there are millions of genes in our body. There are several number of genes in a particular chromosome. We don't have million number of chromosomes in a cell. We only have specific amount that is specific for the species. Okay, so likewise, this P53 is the policeman of the cell. So during this cell cycle, so that is the policeman. Okay, so during this cell cycle, if an unfavorable cell is being produced, this policeman checks it. This P53 gene checks it and destroys that cell through apoptosis. That means they eat their own cell, something like that. They eat their own cell. Likewise, when they are destroying those cells, they have to secrete enzymes. They do not cut and chop cells. They don't have knives. They don't have swords, right? They only have enzymes. Okay, so enzymes are the tool they are using to destroy this cell, to do apoptosis. Okay, apoptosis is when you destroy your own cells. That is apoptosis. Okay, no one else is coming to destroy them. This P53 gene is detecting the unfavorable genes here, unfavorable genes here. Okay, this whiteboard is okay, like this. This unfavorable gene, and the P53 is detecting these unfavorable genes and they are destroying them. So to that, they need enzymes. These enzymes are stored in these organelle called lysosome. Okay, so hope you won't forget the function of lysosome. Lysosome stores the enzymes, lytic enzymes. Lytic enzymes. Okay, lysosome stores lytic enzyme which is needed to degrade waste matter, food particles, dying cells, and anything. Okay, degradable enzymes. So that is the function of lysosome. Okay, so nucleolus, you know, that is the center of center which controls everything. What does ribosome do? What is the function of ribosome? Huh?
Cell cycle regulation?
No, darling. It is in a way regulating the cell cycle, but the main one or the main director of this cell cycle is the nucleus. Okay, so imagine this is the cell. You understood the function of lysosome, right? Do I need to explain it again?
No, I understand.
Okay. If you don't understand any part, please speak up. Okay, I can explain it again. So when you take the cell like this, okay, so there is the main actor here, that is the nucleus. Okay, so this nucleus is controlling everything. So there is one organelle called ribosome. Ribosome. Usually, these ribosomes are attached to the endoplasmic reticulum. So there's a if you know, there is a we call that the system of gutters like thing, that is endoplasmic reticulum. These ribosomes are usually attached to here. Okay, they are like here because when they have a system of gutter, it is very easy to transport, no? So we call, when the smooth endoplasmic reticulum consists of ribosomes, we call it rough endoplasmic reticulum. Okay, so these ribosomes, what they do is, when the nucleus is the main actor, okay, so he is the one who is always sending signals. Okay, so now imagine this cell needs to divide, so he is sending the message, I need to divide. So the message is here. Okay, the message is here. So this message is carried to the ribosome. So like we have hands and legs to do our functions, right? When we need to comb our hair, what we do is we take a comb and comb our hair using our hands, right? When we need to go somewhere, we are using our legs. Okay, we are driving a car, we drive. Okay, likewise, we have our hands, limbs, eyes, and every other parts of our body to do our functions. But cells do not have those limbs. They don't have a hand to do that. But what they use is, they are using proteins. For every function, they are using a protein. Okay, so these proteins are doing the function. So there are messenger proteins called messenger proteins. Okay, there are kind of proteins called messenger proteins. Okay, so these proteins are the people who are doing the function for the cell, like we are combing our hair using the hand. So all those proteins are synthesized, that means produced in this ribosome. So ribosome is a very, very important organelle to the cell body. Why? Because ribosome is synthesizing this protein. So it is producing, so they are getting the message. Now we need to divide. The cell need to divide. The nucleus is telling this. The cell need to divide. So we need to make more and more organelles. We need to make more and more proteins. Okay. So this ribosome is working very hard and giving out proteins. Okay. Different kinds of proteins. So the cell division is mainly an action of nucleus and ribosome. Ribosome's action, every organelle's action is controlled by nucleus. So the ribosome is the main part which produce proteins. So the production of protein is the function of ribosome. Okay. So your answer is correct. The centriole. So by looking at this picture, Itara, you can tell whether this is a mitosis or a meiosis. Why? So, yeah, you can tell this is a phase of a mitosis or a meiosis. So you have here, now in the question they have mentioned that this is the metaphase 1 of meiosis. Okay. This is metaphase 1 of meiosis. So if they haven't mentioned it, you can simply say by looking at this picture without knowing this, you can simply say it by looking at this picture. Why? I'm telling that because if you know, so in my childhood, I used to get confused between mitosis and meiosis. Always it is very painful to me because I know the process of mitosis and process of meiosis, but I used to shuffle those names. Okay, I'm get confused with those names. So I used a easy method to find out that. So I know that one is carried out in the sexual reproduction and one is carried out in normal cell growth. But I am getting confused with the name. So I found a simple way. If the number of chromosome is the total number of chromosome, there is a T here. So there is a T here. So that is mitosis that happens in normal cell growth. So there is no T here. That means the number of chromosome is halved. That is meiosis. That means that happens in sexual reproduction. That is how I memorize those two names. Okay. So hope you don't have that problem. So this is mitosis. This is meiosis. Okay. So in meiosis, what happens? The number of chromosome is halved. The number of chromosome is halved because meiosis happens in production of gametes. Meiosis happens in production of gametes. Gametes occur in sexual reproduction. So when two gametes get together, we can form a zygote. So since this is a revision, I am going a bit fast. If you need me to slow down, please let me know, okay? You can ask anything. I'm very free to answer, darling. So this is zygote. Okay, so when in the production of gametes, the cells undergo meiosis. So in meiosis, the total number of chromosomes are divided into two. Okay, so we have 46 chromosomes in altogether. So every cell in our body, when we take a nail scraping, when we take our hair cells, when we take our skin cells, every cell has 46 chromosomes. Okay, so this number of chromosomes is specific for that particular species. So if you take your hair cell, it has 46 chromosomes. If you take your siblings, any growth cell, it has 46 chromosomes. If you take any human living far away in a different world, if he or she is a human, that person's cell, growing cells have 46 chromosomes. That is the identity of a species. If you are having 46 chromosomes, you are a human. Okay. So likewise, the animals have elephants like 70 kind of number of chromosomes. The different species have different number of chromosomes. Okay. So now as humans, we all have 46 chromosomes, and that is a constant. That is a constant. Okay. So this 46 chromosome, when they are undergoing mitosis, remember mitosis has a T, so there the total number of chromosomes are going to the next generation. Okay. So when they are producing new cells, those cells also have 46 chromosomes. But when they are undergoing meiosis, they are having only 23 chromosomes. Why? Because they are producing gametes here. They need to take one gamete from another human. Okay. So when they collect together, they will make the total number. So for that reason, they have to half the number of chromosomes. So by looking at this cell, we can tell, so where's that cell? Okay, by looking at this cell, we can tell whether this is undergoing meiosis or mitosis. Why? When we take the cell, if it is undergoing meiosis, mitosis, all the chromosomes are lining in the center this way. Okay. So when the centriole is here, they are producing threads like this. Okay. So these threads get concentrated. So they are getting detached. So these chromosomes are getting detached. These chromatids of a one chromosome is getting detached. So this is mitosis. So in a mitosis, you won't see this picture, darling. In this picture, these chromosomes are lying pairs like this, like this. They are not in this line. They are in two lines, like this. See, they are in two lines. If this was a mitosis, they should be in one line like this. Okay. So by seeing at this picture directly, we can say that this cell is undergoing meiosis, not mitosis. Okay. Because they are lining in this cell in a different way. If it underwent mitosis, they should go in this way. The one chromosome should be in this way, and the other chromosome in this way and this way. So when they have chromatids like this one is connecting to these chromatids, and they are pulling that away. So these are getting separated, and each chromatid is moving into different box. But here, that doesn't happen. What happens is, both of these chromosomes are paired. So, there are only 23 pairs. In mitosis, the center has 46 chromosomes lined in a line. Okay. So this question is based on meiosis. So, the next question, explain why it is possible for chromatid A to be genetically different from chromatid B. Can you please tell me your answer, darling?
It can be DNA replication.
So in DNA replication, what happens? DNA replication, what happens? That gives rise to genetic variation. So, Tara, what happens here is ideally in a normal cell, in a cell which is in a normal condition, we only have this part, okay? We only have this part. We only have this part. So when it is getting prepared for this replication, that means cell division, they are forming another copy of this chromatid. They are dividing, the DNA is dividing, DNA is replicating, and they are making another copy. Okay. So this copy has to be ideal. If it is the actual situation, ideally similar to this. Okay. The first one is that. Okay. So if it is an ideal situation, this limb, this arm is equal to this one. Okay. And this arm is equal to this one. Okay. So now the question here is, now this arm is not ideally similar to this arm. This arm is not ideally similar to this arm. This chromatid A is not similar to chromatid B. So when they go on to two different cells, this is chromatid A and this is chromatid B, they are giving rise to two different kinds of cells. That means, so hope you understand what I'm saying. So ideally they should be equal. They should be similar, but it is not like that. Why does that happen? There can be two reasons. I'm telling you the first obvious reason. Okay. So they are they are replicating and making double. They are being doubled and being organized in the center of this cell. Okay, the cell line, they are lined up here, and here you can see when they are getting lined up, So for an example, darling, when they get lined up, we have 46 chromosomes. Now they have 23 pairs here. Okay, there are 23 pairs lined up in the center. Okay, so this chromosome is a paternal chromosome. That means a chromosome that is coming from the father's side uh, to control something. Imagine the control the skin color, and there can be several features. Imagine, just assume this is the paternal chromosome responsible for the skin color. Okay, so it is pairing up with the maternal chromosome, which is responsible for the skin color. So it is not pairing up with some a random chromosome which is controlling some other thing. It is pairing up with the same chromosome which is controlling the same feature. Okay, so now there are 23 chromosomes finding their partners. They have found their partners which has come from the paternal side. The paternal side chromosome is paired up with the maternal side chromosome. Okay, so now both of them are responsible for the color of the skin. Okay, now clearly look at here. When they are pairing like that, they can get crossed over. So this usually happens when they are detaching from each other. They get earlier they get strangled here. Okay, they get connected here, and when they are detaching, when they are pulled towards the centrioles, these detached parts are getting mixed. So they are the maternal chromosome is getting a part of paternal chromosome, and paternal chromosome is getting a part of maternal chromosome. So we call this crossing overs. Okay, so you can clearly see, Tara, here they have showed that there is a crossing over. See, this color is changed. They are having two multicolored here. Okay, by the color is changed. This light color one indicates one type. Imagine that is maternal type. This maternal type has gone to this part. That means a crossing over has happened. So the most obvious incident here, which has given rise to the genetic mutation, is the crossing over. So let me explain that again, darling. Ideally, this limb has to be similar to this limb, exactly similar to this limb. Ideally, it should be 100% similar. But since this crossing over has happened, this limb is getting a part of paternal chromosomes here. So it is having a part of paternal chromosome here. So now this limb is not ideally similar to this limb. So the two cells which are forming are not similar. That is the obvious answer to this question. So if we move a much deeper, we can tell another reasons why there can be genetic mutations. I told you, when there is a normal cell, there are only 46 pairs of single chromatids. So when they are ready to replicate, what they do is they form the exact copy of that same chromatid. That means they are pairing in two, like this. Okay. So the whole part is called as a chromosome. In meiosis, darling, the two chromosomes are pairing, but here this is chromatids. Okay, not chromosomes. They are chromatids. So this one limb is ideally similar to the other limb. Okay, so they are ideally similar. But when they are replicating, DNA is something like this, no? DNA is something like this now. Okay. So they had two limbs like this. Now only they have two limbs because they have replicated. Okay. So uh, they are formed of these small units. Okay. So when they are replicating, there can be mutations. Okay. So there can be mixed nitrogen bases. You know they are made out of bases. Okay, nitrogen bases. So there can be mixed mutations. There can be uh, there can be repeated mutations, mixed mutations when they are replicating these. There can be functional errors. No, these nucleotides are not joining ideally. So nitrogen bases are not joining ideally. They are getting mixed. So there can be genetic mutations. There are different kinds of genetic mutations. Point mutation, the point mutations and triplet mutations. There can be, you don't have to remember those things. There are point mutations. That means a single point is different. So when they are doing that work, they are missing some point and they go wrong in that point. So what happens? Ideally they should be equal, but they are not because of a functional error. They are not meant to be different. They should be ideally 100% similar to each other, but due to a functional error, they are not similar. So if they are not similar, what happens? The two cells made out of each of them is not similar. So there are two answers for this question. The first one is, this is obviously having a crossing over. That means when the chromosomes are pairing, they get the parts of the chromosomes get mixed. That is the crossing. Okay. And the second answer is, there can be genetic mutations. If to add more marks, you can name one genetic mutation like point mutation. There can be genetic mutations which can happen in DNA replication. So the DNA replication is a different process. The detachment of the chromosome cell division is a different process. So there can be crossing overs happening in cell division and DNA mutations happening in DNA replication. So both of these factors give rise to genetically different cells. Okay, chromatics. So that is why there can be genetical mutation. Did you understand that, Chitara? Do I need to explain it again?
I understood this.
Okay. So let's move. Okay, the next question. Okay. So did I show you the next question? No. Here. Here, try the next question, darling. Okay, Tara. Do you know the phases of meiosis, cell division? Can you name them?
I didn't hear properly.
Can you name the phases of meiosis? Can you name the phases? So the meiosis consists of two main phases, telophase 1 and telophase 2. So there are three phases in telophase. Do you know them? You can't remember. You can't remember, then it's okay. It's okay. Do you know? Oh, you can't remember.
I think I can't remember.
Okay, that's okay. That's perfectly all right. So we are talking about meiosis today now. So this is meiosis. Meiosis happens in production of gametes. Okay. So in meiosis there are two main parts. That is telophase 1 and telophase 2. Okay. In telophase 1 there are three main phases. Okay, there are three main phases. One is prophase. That is, the second one is metaphase. The third one is anaphase. Okay. So in prophase, what happens is when this is the if this is the cell, I told you in general, Itara, in general, if we take the nucleus, we cannot find chromosomes like this, a specific, very neat chromosomes. There are no, they are not organized like this. It is like a very messy place, okay, like this. So when it is near to the cell division, what happens is they get condensed and they make that original chromosome like this, okay. So now there was a chromatid. So this chromatid, we call them chromosome at that time. But now, since they are near to the cell division, what they do is they replicate and make an exact copy of themselves. So now there are two chromatids in one chromosome. So they are connected with a with a point called, oh sorry, where was it? Okay, here. It was connected with a point called centromere. Okay, this point is called as centromere. Okay, so now we call the chromosome to a unit consisting of two chromatids. Okay, so in prophase, what happens is these chromosomes are getting condensed and they are replicating, they are getting condensed in this telophase 1. So since this is happening in telophase 1, we are calling this as prophase 1. Okay, in prophase 1, these chromosomes are arranging in pairs. So we call them homologous pairs. Okay. One of these pairs is called as homologous pairs. Why? Homo means that means the same. Same. That means homogeneous solution. That means the consistency is same. That means these two chromosomes are controlling the same feature. Okay, one is from the paternal side, one is from the maternal side, but they are for the same feature, for this for the controlling of the skin color, for the controlling of the nature of the hair, the Likewise, for the height of the height of the baby or the person. So for that specific feature, they have chromosomes. So they have 2 chromosomes to control one feature. Okay, they have 2 chromosomes to control one feature. That means one from paternal side, one from maternal side. So those pairs, those chromosomes get paired. We call them homologous pairs. Okay, homologous pairs. So now 23 homologous pairs are paired in the center of this cell. Okay, so we call this as prophase 1. Okay, so in the meantime, centrioles are moving to the either side of the cell. Okay, the centrioles are moving to the either sides of the cell and they are sending these threads. Okay, so now they have sent these threads. They have been attached to these chromosomes. Okay, these chromosomes. So this is called metaphase. Since it is happening in the telophase 1, we call it as metaphase 1. Okay. So when they are getting, these threads are getting shorter and shorter, so they are pulling these sets of chromosomes to the either sides. Okay, so they are accumulating near these centrioles. Okay, like this. Like this. So this is the cell. So this is called anaphase. Okay, at the end of the anaphase, the cell is dividing, the cytoplasm is dividing and making 2 cells. So there are prophase, metaphase and anaphase. So I told you there are the structure which connects 2 chromatids. Okay, the structure which connects 2 chromatids together is called as centromere. Okay, so tell me, Tara, does centromere separate in metaphase 1? Does it get dissociated, separated in centromere 1? Sorry, in metaphase 1.
No.
Very good. It does not get separated in metaphase 1. Why? Because when these threads are pulling, the whole set of chromosomes are moving to the 2 poles. The chromatids do not get separated in metaphase 1. So the centromere is being intact. It is not separated. But in metaphase 2, it is getting separated. But we will be talking about metaphase 2 the next day. So I hope this is too much for you today. So is there anything you did not understand here, darling? Or do you need me to repeat them? I'm talking about the whole thing. Do you understand about the cell cycle, or do you need me to teach a bit about cell cycle the next day? Or shall we move on with the questions and discuss the parts related to the question? What do you prefer, darling?
I understood.
Okay, you understood today. So do you prefer this way or do you want me to recap the unit again and move on to the questions?
I think it's better to do question.
Sorry, darling, I didn't hear you.
I think it's better to do questions.
It's better to do questions. Okay, we can talk the theory parts in between questions like this. Okay, they're related to the questions. Okay, you may have done the theory earlier. Okay, so we will do likewise. Okay, we will discuss questions. So I will finish. I'm focusing mainly on the latest past papers. So I'm so the discussions will be based on the past papers. So I'm first of all I'm doing unit wise past papers. So we will continue this unit the next week also. I will do few questions from the past papers regarding that unit, and we will move on unit wise and complete the units first, and we will do full papers with the timings after that. Okay, so you have to read around this unit. I'm maybe it's unit 38B, I think, regarding cell division, mitosis, and meiosis. I will discuss small parts related to the question. Okay, uh, so talking about the questions, talking about the exam, darling. So, do you know Michael Jackson? Who is? I hope you know Michael Jackson. He's a very good singer and a very good performer on stage, right? He's a very excellent performer on stage and a well-known, world-renowned dancer. Okay? So, even though he was a very good performer, he always practiced 100 times before a performance. Before a concert, he used to practice it 100 times on that particular stage. Imagine he is going to Melbourne and doing a concert on Melbourne stage. He's doing that performance 100 times before the concert date. He used to do that when he was young because practice makes someone perfect. Okay, practice makes someone perfect. When you practice, it goes to your spinal level. That means it becomes a habit. Imagine, I hope you don't have the first hand experience. When you are driving a vehicle, first in the very first time you are thinking about, now you are pulling the handbrakes, now you are putting the gear, now you are pushing the clutches and all that, pushing the brake, and you're putting you're putting your foot on the accelerator. That likewise you are thinking every step when you are driving a vehicle for the first time. But when you get used to that, when you practice it several times, with the time, it becomes a reflex. Now you don't know, when you come home, you don't know whether you have actually put on the gear, but it has spontaneously come to you. Likewise, when you brush your teeth, you don't think, oh, now you have to put on the signals to the brush. Now you have to brush your teeth. That we don't do that now. No, we did that when we were very small. Now that is happening spontaneously, and washing mouth after that happens spontaneously. That means we even can't remember whether we brushed our teeth or not, because that happens very spontaneously through our brain. So likewise, if you practice something, that becomes spontaneous, that becomes a habit. So practice makes someone perfect. So you have to practice papers, you have to practice questions that makes you perfect. Through that you can excel in this field. Always study based on questions. So you should not only study, you have to look at the questions and go back to your book and find out what you are missing. Okay, so you can try them. You have to read the unit again. We shall practice this again and again. That is why I am reminding things again and again, because when you are hearing things, it is become very familiar to you. Okay, so we will discuss another. We will do few more questions next day, since this is the first day. I discussed only one question. We'll try to discuss more in the next day. Okay, it doesn't have to be the next Saturday. I can do any time you are free. So it can be about twice a week, thrice a week. That doesn't matter. If you are ready, please leave me a message. I will arrange a time and we'll do the questions. Okay? Is that okay with you?
Yes, teacher.
Okay, and have a nice day, darling. We'll stop here.
Thank you, teacher.
Okay. Bye. Good night.